Hello boys, welcome back to another interesting session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 18th September 2020, and we have been, as you all know, discussing about biotechnology and its applications. One of the best applications that we need to discuss is Hybridoma Technology. This comes under gene therapy. So, in this, we will be talking about monoclonal antibodies. That is what we are going to discuss. Monoclonal antibody is a specific antibody produced from normally short-lived antigen activated bone cells that has been immortalized by hybridizing it with myeloma cell by hybrid cell culture technique known as Hybridoma Technology. That is Hybridoma Technology. In this technique, the antigen against whom the antibodies are required is injected into a rat, as you are seeing. Injected into a rat or a mouse. After some time, B lymphocytes from spleen of an animal are isolated. B lymphocytes are isolated and mixed with myeloma cells. So there's a mixture. These myeloma cells are isolated from cancer cells of bone marrow. Antibodies producing B lymphocytes and myeloma cells fuse together to form hybridoma. This is what the technique is. Here there is a fusion of two cells, and we are using the negative property of proliferation, cancer cells, for good purpose. So basically, one way the body's immune system attacks foreign substances is by making large number of antibodies. An antibody, as all of you know, is a protein that sticks to a specific protein called antigen. Antibodies circulate throughout the body until they find and attach to the antigen. Once attached, they can force other parts of the immune system to destroy the cells containing the antigen. This is the work of antibody. Researchers can design antibodies that specifically target certain antigen. So we are going for specificity, such as one found on cancer cells. So this technology is mainly to help cancer patients. So once they can make cancer cells specific antibodies, then they can make many copies of that antibody in the lab. These are known as monoclonal antibodies. This is the purpose, monoclonal antibodies, and we abbreviate it as MABS. So you need to write a small m and small m, capital A, and BS small. So this is how we write, the scientists write MABS, or it also can be written as MOABS. Monoclonal antibodies are used to treat many diseases, including some types of cancer. So that is the purpose. To make a monoclonal antibody, researchers first have to identify the right antigen to attack. Finding the right antigens for cancer cells is not always easy, and so far MAbs have proven to be the more useful against some cancers other than others. Some monoclonal antibodies used to treat cancer are referred to as targeted therapy because they have a specific target on a cancer cells that they aim to find, attach to, and attack. But other monoclonal antibodies act like immunotherapy because they make the immune system respond better to allow the body to find and attack cancer cells more effectively. So basically what we do in this is the fusion. So how do you do that? In the first one, immunization. So we must be thankful to two scientists, Kohler and Milstein. Thanks to these gentlemen, Kohler and Milstein, in 1975 devised a technique for preparing monoclonal antibodies. Since direct biochemical purification of monoclonal antibodies from polyclonal antibodies is not possible, this technique is called Hybridoma Technology. That's what we said, Hybridoma Technology. The process involves the fusion of normal activated antibody producing B cells, bone cells, with myeloma. What is this myeloma? Cancerous plasma cell to produce a hybrid cell called hybridoma. Hence it is appropriately named as Hybridoma Technology. So what is it we are doing here? First step, immunization. A mouse is injected with a specific antigen against which antibodies are to be generated. You can see here specific antigen is injected. And what are those epitopes? Epitopes are the areas where the specific antigen antibody can sit and make the antigen to get blasted or the cell get blasted. So that is epitopes. And once the antigen is injected into the mouse or rat, naturally antibodies start producing. So isolation of serum and spleen cells, second step. Polyclonal antiserum containing a mixture of monoclonal antibodies, each specific for different epitope. Epitope is area of an antigen is isolated. So you can see here the specific antibodies. And spleen cells are isolated, which comprise of bone cells, each producing specific antibodies. And next, in step number 3, what we are doing? Clonal selection. When an antigen reacts to the cell surface receptor of a beta lymphocyte, it proliferates rapidly to yield a population or clone of bone cells producing antibodies of the same specificity. That's why epitope. Antibodies of only desired specificity are selected. They are specific to only one antigenic determinant. One such plasma or B cell for each is selected. As you are saying, see, antibodies are produced, normal plasma cells capable of producing specific antibodies, and HgRAT, that's enzyme, okay? And then in fourth, what we aim at, fusion. The plasma cells and the myeloma. See, here we have separated plasma cells and spleen cells, separated in step 2. Now the plasma cells and myeloma cells are mixed together. Some of the plasma cells and myeloma cells fuse to form hybrid cells. And these hybrid cells are now capable of growing continuously in culture while producing antibodies. So you can see here unfused plasma cells, unfused myeloma. But here you can see at the bottom fused hybrid cells which produce the antibodies. And now selection of hat medium. The mixture of cells in hat medium, step number 5 I am talking. The mixture of cells transferred and incubated in a selective hat medium that allows only hybrid cells to grow. You can see here hybrid cells are growing. Unfused plasma cells can't survive, they are dead. Myeloma cells cannot survive, dead, but whereas hybrid cells are surviving. And the hat medium refers to hypoxanthin, H, A for aminopterin, T for thymidine. That's how the HAT. So in that medium, the mixture of cells are incubated. And once that is done, look at that, hybrid cells. Hybrid cells with HgRAT enzyme survives, produce antibodies and grows successfully on hat medium. Whereas this is dead, and this is surviving. And now proliferation. Once we get that, the nature of cancerous cells are proliferation, means increasing uncontrollably. That property, negative property, we are using for positive purposes. So hybrid cells proliferate into clones called hybridomas, and these are screened for production of the desired antibody. Now you can see the hybridomas, process of immortal growth, properties of myeloma cell as well as the antibody producing properties of bone cell. Thus, the resulting hybrid cells secrete large quantities of monoclonal antibodies that are cultured indefinitely. Now, screening and production. Seventh step. The selected hybridomas are then cultured to produce large amounts of monoclonal antibodies. Look at this, they are very, very specific hybridomas. The antibodies with specificity are cloned. You can see the cloning of this. So, this is how the monoclonal antibodies are produced. These are produced by a single clone of cells. Therefore, MAB, monoclonal antibodies, MAB means, is a single pure type of antibody. They can be made in large quantities in the laboratory and are increasingly coming into use as therapeutic agents. So, main purpose of this is to cure cancers. So, the monoclonal antibodies that are produced will target the cancer causing antigens with the epitopes. They can sit and break the cancer cells. So this is one of the best gene therapies that modern biotechnology scientists are using, and this is a great boon for all the patients who are suffering with varieties of cancers. And such experiments, I am sure, will continue, and the humankind is going to have lot of fruits of biotechnology. That is the reason